what I thought I was tasked to do and what I've decided to do may not be the same things, but what I'm going to do is talk about some of the technologies that we have in the core, and I'm going to start with, just short read, and then I'll head into the more complicated and newer ones, such as the spatial transcriptomics that we're doing. My impression is that most of you are in the analysis part of data, and some of you may or may not know what a flow cell is and how we get the data to the flow cell and then send it to you. So please ask questions. I have a lot of slides, but I'm not afraid just to, skip a bunch. If you just say, this is really redundant, Melanie, I'm done with this, okay? So, so the first one, and one of the ones that we've been doing the most, and we still are, is our short read technology, which would be primarily on our Illumina platform. And so the generic workflow would be that we have sample preparation. It's also called making a library. We then quantify it. It gets clustered on things like this, which is a flow cell. We sequence it, and then you guys can figure out what it all means by doing the analysis part. The positives of this particular technology is that it's a high output. So this particular flow cell right here has 10 billion clusters that are available for reads. So it's 2.5 billion per lane. So it's a lot. The number of projects we put on here can be as many as 20 per lane. It's a logistical nightmare sometimes. But anyway, we're pumping out a lot of data. So I don't know how much of it you all are analyzing, but we're doing a lot. It has a relatively low error rate and low amounts of DNA requirements or RNA requirements. Some of the cons are that it is relatively short read. So if you have repetitive regions or something like that, then you need to find another technology. There is also some PCR bias, and that's just inherent in this particular technology, and I'll show you why in a few minutes. So this is the overview of the library construction. And if we're starting with the RNA, then we're going to make cDNA first. So looking at the DNA fragments, we do a repair and then do what we call an A-tailing, where we'll adenylate the 3' end. Typically, we're ligating on adapters. So the adapter has a free T, which makes the ligation much more efficient. And then we have a library that's got an insert with these adapter sequences. So what are these adapter sequences? And they have specific sequences for binding this fragment to the flow cell. They also have binding sites for primers, and they also have indexes. So if we look at the structured details, and I'm going to minimize that. If we look at the structured details, here's our sequence of interest. This P5 represents a sequence that's complementary to an oligo on the flow cell. This P7 also represents a sequence complementary. And then this R, okay, let me find where my arrow went. Okay, never mind. My read 1 sequencing primer is in the red. The read 2 sequencing primer is in the upside down, and then our index. So this represents what would be a single index library. Most of the time now we're doing dual indexes. So there's another sequencing primer on the P5 end. So why do we need these? So this particular slide, basically this flow cell is like 2 slides put together. So our libraries are going to flow through this flow cell, and we have oligos that are on the surface, on the top and the bottom of each lane. So what happens is that these libraries—
Can we pass these around?
Sure. So if you look down, okay. Oh man, I can't find where my arrow is. What's an easy way to find your arrow?
So if you hit Control, excuse me.
Okay.
There you go. You have
Oh yeah, that's perfect. Okay. So if we start down here on the bottom left, this represents our library that's got a P5 end as well as a P7. It then hybridizes to the correct oligo that's on the surface of the flow cell. And the chemistry uses that oligo as a primer. So it now makes a complementary template that's covalently attached to the surface of the flow cell. And this is where the bridge amplification comes in, is that because we have this other end up here, it just bends over to hybridize to another oligo on the same surface of the flow cell. And it uses that oligo as a primer and makes another copy. So what happens is that this process gets repeated about 35 times. So what you've done now is that from your single template, which is right here, you've now generated a cluster. And this is where we use the word I prefer, like when I'm talking about what's on this lane and what the capacity is, I like to say that it's got the potential for 2.5 billion clusters as a way to identify what it is that we're doing. So this is where you have PCR bias that can potentially come in, is that not only is it when you're making the library, but then it's also on the instrument, okay, is generating this cluster.
So you're saying you have clusters of different sizes?
So the cluster is going to be, if I go back, okay. So this cluster here, each cluster is going to represent originally from one single template. So if that template, let's just say your insert size was 350, okay, that would represent, a cluster would be 350. Other libraries on the flow cell might be 450. So the sizes will go wherever that cluster has been generated. The other thing that happens is that you can imagine, because we're doing this bridging all the time, one of the things that the final step is that there's a cleavage step so that all of these templates are in the same orientation, because you would have had complement, reverse complement, complement, reverse complement.
Sorry, Melanie.
Yeah.
Can you, I didn't quite understand the PCR amplification bias thing. So at the end of the day, are you saying that it's more likely that you miss shorter reads, or what's the?
No, it's just that PCR, this PCR amplification could introduce some mutations. So it might be substitutions, it might be, and it's not going to be on all of them, okay? If it happens early on, then it might be, okay? But this is where anytime you're doing PCR, there's going to be some bias, okay? So what's the point of the clusters? And I really like this diagram here, because after we have the clusters, we're going to hybridize a primer, okay? And once that primer has been added, then we have all of these fluorescent nucleotides. And it will add one fluorescent nucleotide and then stop. It's actually blocked from progressing beyond the first nucleotide. So with the cluster over here, and after you've hybridized this read one primer, it's going to add a fluorescent complementary base. And it's now, because it's a cluster and it's about 1,000 copies, you've got a fluorescent signal that you can actually see. So ideally, if you were thinking about, like, improving technology, you would want to do it so maybe it would be more sensitive than having to amplify this much, okay? So for this particular cluster, it was adding a G. This particular cluster, and again, this is from a single template to begin with, all right? It added a T, a C, an A. So in the first cycle of imaging, for instance, if we look at it, this down here is looking at a tile on the flow cell that's got a number of clusters, okay? And so in the first cycle, for instance, where number 1 is, it was green, so it's going to be a T. So after it does the first cycle, then it cleaves the blocking that was on that base, and it adds the next nucleotide. So that's what we call cycle 2. So when on the order form, when we're asking you, how many cycles do you want to do? This is how many iterations of adding a base do you want to do? So we're going to, in this circumstance, the same cluster here in cycle 1 with a T, in cycle 2, it added a G, okay? And so this is the sequencing by synthesis, which is how the Illumina technology works. So this,
Sorry, I'm
yeah,
sorry.
go ahead.
Can we go back one second? So this is really interesting. So when you say when you ask the question how many cycles you do, so is that what sequencing depth is, or is that something else?
No, sequencing depth is where I would say to you, how many clusters do you potentially want? Okay. So for instance, if you're giving me an RNA-seq library, if we're going to sequence RNA, all right, typically, if we're doing bulk, I will suggest that the minimum amount of reads should be 20 million. And usually it's what I mean by that, it's 20 million and it's going to be paired in 100 reads. I'm putting everything as much as possible on this flow cell in the core. And the reason why is that Illumina gives us a 40% discount on this flow cell only, okay? The other ones have a discount, but not as much as this. So this is the cheapest sequence, the money per base is on this flow cell. So like 20 million reads is probably gonna be, I don't know, 34 bucks, okay? So this is why. So you may come to me and say, I only want single read. Yeah, you're gonna get paired in. Just don't take the extra paired in read if you don't want it, all right? Because it's just a lot less expensive for you. We could do single read on this SP, or S1 or whatever, wherever the SP is, but it's going to be a lot more expensive for you.
And so again, just to make sure I have everything straight. So when you're talking about read depth, that's basically translates to number of clusters.
Yes.
Is that correct? Okay.
Yes.
Okay.
So, some people will say, so for 20 million, that's going to for sure give you your high expressors, meaning that those transcripts that are in higher abundance, we're going to have more of those, you're going to see more. If you came to me and said, hey, I've got some low expressors that I'm worried about, then it's like, okay, we need to increase the depth and see if we can get more. So we might go to 40 million. All right, then you're spending $68. All right. And so, or if we're doing, that would be for messenger RNA enriched, if we were doing total RNA, meaning that we depleted all of the ribosomal RNA and everything left is poly A or long non-coding or whatever, then we might want to sequence more too, maybe up to 50 million. So this is where, when we talk about read depth, this is where what we mean by read depth. Or if we're talking genomic DNA and you're coming to me and I've got a human sample, I want 30x coverage. So then I'm going to figure out, I'm going to do a calculation where I take, okay, 3 gigabases times 30, all right, gives me the number of bases. And then I divide that by the number of bases we're going to sequence to get the number of reads. And it's about, I think, 500 million, something like that.
Right.
Yeah.
So about the cycles,
Yeah.
is there like a recommended, or how do people get to decide, or is there a minimum?
So that's a great question because typically, we're going to do paired in 100 for sure on this. But you can also think about it that the number of cycles is, if we're doing paired in 100, it means you've now got 200 bases. Okay. So if your insert size is only 100 bases, then that means on the paired in read, you're resequencing the exact same thing that we sequenced on the forward read, right? On read 1. So where people would want, for instance, paired in 150, which is what we can do here, then I'm gonna advise them to make sure that their insert size is big enough that you're gonna probably get some adapter sequence anyway, but to get the most bang for your buck, you want your insert size to be at least 300, okay? And sometimes for genomic assemblies, they actually like to sequence over a little bit. And so, so that read 1 and read 2 will kind of repeat some bases in the middle. And so, but we're still looking at something that is at least 300. And keep in mind that when we add on those adapters, they're at least 60 bases. So that when we're looking at the size of the library, we're talking about your DNA, your insert size, as well as your adapter size.
If we go beyond our insert, is that just kind of like?
It just means that you need to do some trimming. Okay, so you would want to trim off your adapter because it's not going to align. Okay, and most of your fast QC programs will tell you this is how much adapter sequence you have. Okay. Yeah.
Question here.
Uh-huh.
So if you are running 35 cycles, meaning from one end, let's say read 1, you are adding 35 nucleotides.
That's correct.
And then typically if you have read 2, meaning that your insert size only added around 70 nucleotides. And if it's longer than that, so there's a gap, right?
There is a gap. So some people don't care about the gap. Let's just say that they're doing, RNA-seq, okay? They want to be able to align those 35 and the other 35. And as long as they can align those ends, then it's like, okay, I'm going to call this, beta-actin or whatever. Some people say you need more than 35 bases because you could potentially get alignment at other places along the genome or the transcriptome. And so this allows you to have more confidence, when you have bigger or larger amounts of sequence on either end. But there are some, that will say, you could get by with 10 million and only sequence 35 or whatever. I don't feel like I can recommend that to you. You would need to come to me and say, I don't care what you say, this is what I want. And I'll do what you want, okay? I will do what you want, but I can't recommend that. And so I usually say that a minimum of 20 million. So this is what a FASTQ file looks like. You've probably seen this before. The interesting thing would be, this is the flow cell ID right here. And this is what lane it was on. So if we have 4 lanes, it would be 1 of 4. And they're labeled by a tile. So this lane has tiles all the way down it on the top and the bottom. And so it's going to tell you which tile that cluster was on. And then it's going to tell you the actual coordinates of where it was on that tile. And then the barcode. And then these would be your read bases. And then this would be your quality score down here. Okay, or your Fred score. Yes.
You know if there's a purpose of the third line of FASTQ? I've wondered this for as long as I've been.
You know what? That's a question you should answer and tell me. I don't know.
Yeah, they teach it in the class that I TA for as the placeholder. And I'm like,
Okay.
I don't know what that means.
I don't know what that means either. It's a great question though.
It just seems like it's taken up space to me.
I would,
The separator?
Yeah.
the separator, and it's only in, it's in every line. I don't know.
4 lines? Maybe it's to make it 4 because 4 is better number than 3.
I don't know. It's a good question.
Melanie, could I ask you a question about lanes? So when we submit for sequencing, I think we, correct me if I'm wrong with this, but I think we ask for a particular number of lanes. Is that
You can. So for instance, the way you would do that is, and actually on iLab right now, we're just asking for how many reads do you want? So let's say, for instance, you say, I want 5 billion. Okay, that's going to be 2 lanes of this. But a lot of times people don't know how many we can get per lane. And so we're just asking you now, how many total reads do you want? And it may be like 1.5 lanes. So the reason why we dual index is because you can't afford just 1 lane. Okay, I mean, you could, but if you only need 100 million reads and this thing is 2.5 billion, you're going to come to me and say, I just want a partial lane, you know, and how are we going to do that? So when we make libraries in the core, and what we recommend and request that you do if you make libraries is that you put on unique dual indexes, so that when we demultiplex this lane, we can have confidence that you're getting just what you asked for, okay? Now, sometimes we have a problem. We had a problem in that somebody didn't provide us their correct barcodes. Rarely does it happen, but it does. So in that case, then I had a client that was getting reads that weren't theirs. All right. So then I have to go back and figure out how I'm going to solve the problem. Okay. And most of the time, in one instance, I went back and said, you need to pay for the resequencing we had to do because your postdoc didn't give us the correct barcodes. All right.
Yep.
So, and you know, the barcodes are complicated. All right. And it's because aluminum makes it that way. So for reading the barcode on the I7, the P7 side, it's really easy, is that after you've done your 100 cycles, that template is removed. And then they hybridize another primer and they start through the index cycles. Okay, that's okay. But for the I5, I mean, for the P5 and for the read in, it repeats the bridge amplification and regenerates that template strand. So now it's in the opposite orientation. So then it binds the primer for the read 5, okay? I mean, the I5 index. Well, they've changed their chemistry. So, you know, at one point it was when they changed it from version 1 to version 1.5, we needed the reverse complement of that I5 barcode. So now people need to keep track. And, you know, honestly, if you drop off libraries or you do your iLab form, you're not going to know which version of chemistry am I doing, you know? And so we just ask now, most of the time, I think, that you provide us both sequences for your I5, and then we'll figure out which one it's supposed to be for you. But yeah, it's I appreciate y'all listening to that little part because it will help. You can have some sympathy for on our end, trying to multiplex all of these libraries and trying to make sure that we've got the correct barcode sequences. So what are we using this short read for? We do it a lot for whole genome sequencing. The cost has come down so much, you know, that we can do whole genome at 30x for less than $1,000. And so it's really made a big difference. We do a lot of exome sequencing, people that don't want to pay for whole genome. Whole exome is, you know, right now we're doing it and it's a 39 megabase region, you know, that represents exome, human exomes. So you don't have to, it cuts down the cost. So for human exome, for library prep and for sequencing, it's about $200. Okay, so that really puts it much into an area that's pretty affordable. We can do targeted sequencing. So most of our targeted sequencing that we do is actually 16S, people looking at, you know, microbiome type work. RNA sequencing, we also do some circulating tumor. So these are some of the applications that we're seeing with short read. Some of the challenges, yeah, I've already mentioned, you can't get through all these repeated regions, you know, with a degree of confidence. You don't get haplotype information. These sequencing libraries depend on enzymes. So again, talking about some PCR bias. RNA-seq libraries, it's bulk. It represents a lot of cells. Human genome is very large. And so if we're going to do like a trio study, you know, if you've got parents as well as an affected child, that gets expensive. And so still working on that. So we have long range technology to try and deal with some of those issues. And in the core, it's using PacBio And so in my next bag of tricks, let's see here. Yeah. Smart cells look like this. So for smart cells, a lot of the work we do is iso-seq. And so total RNA, we make a smart bell template, and I'll show you a few minutes what that looks like. We also do some structural variant detection, okay, using PacBio. People that are doing whole genome assembly would like to do PacBio because we can get these longer reads. So what does this look like? PacBio is very different than Illumina. For one thing, we are involved in making the smart bell templates, and there's no clustering so that you don't have PCR there either. What happens, I call it a dumbbell, but they're labeling it a smart bell. So this is our DNA, this is our smart bell template, and our insert. We ligate on these hairpin loop adapters on both sides, and then we anneal a sequencing primer. We bind polymerase, and then we put it into the instrument. So in the instrument, this is what's called a zero mode waveguide. And this polymerase bound template sits in the bottom of this zero mode waveguide. There's a laser that's shining on the bottom, and you've got these fluorescent nucleotides in solution. So the fluorescent nucleotide that is complementary binds into the active site. So what's the nice thing about PacBio is that this is taking a movie. So rather than sequencing by synthesis, okay, this one is an ongoing movie. And the camera is looking at what fluorescent dye is sitting in the active site of polymerase. So it's looking at that fluorescent dye, seeing how long it sits there before a different fluorescence comes in. So in PacBio technology, whichever base is coming in, there's a certain kinetics of addition so that it sits in that active site for a unique time. So the software is looking at how long is that particular fluorescence there in the active site, and then we're going to call it an A because of the amount of time. So right here, this represents what would be, for instance, if the C nucleotide was sitting in there, this would be the pulse of time, and then an A over here. So it's an ongoing movie. So the movies can be up to 30 hours long. It only does one of these at a time, and this one has about 8 million zero mode waveguides. It's a Poisson distribution. So if we're lucky, we're going to get maybe 50 to 75% populated zero mode waveguides. This technology is also known as having a higher error rate. The nice thing about it, and some of it's just because, you know, you've got these laser light pulses, which eventually the enzyme's going, yeah, no, this is not good, right? But because you have this smart bell template, it's sequencing around and around. So because the errors are random, the more time it goes around, then it increases the accuracy of the read. And because what happens is that if you were to linearize what this would look like, these represent subreads, and these get collapsed into a consensus read, which is the higher accuracy. Okay. In terms of insert size, it can go anywhere from, you know, a minimum of 500 base pairs. If you go any smaller than that, it's not going to be able to go into, it's not going to be effective in that well. The really long ones have a hard time getting in, so we have to load more on to try and force it into there. And you can also detect modified bases because you've done no PCR. All the modified bases are still present. So the nice thing about the modified bases is that, again, if it's methylated A, for instance, that is a different length of time that it sits in the active site than the unmethylated A. And so that's pretty cool. So some of the challenges for this is that it needs a lot more sample. So for us to go into, and they're getting better, there are these low input, but the standard protocol is 5 micrograms of sheared and concentrated DNA. Sometimes people can't give you 5 micrograms, all right?
And what is it, just for comparison, what is it for the alumina?
For alumina, we can go down to sub-nanogram
Wow.
amounts if really needed to.
So there's at least 3 orders, man.
Yeah, we prefer higher amounts
Sorry, yeah.
because I don't like to give you all a lot of repetitive a lot of repetitive reads, right? Yeah,
A question on the plus side, methylated,
yeah,
that is, the site is methylated, so
Yes.
it means SmartSeq can detect methyl sequencing
yes.
by sulfur, kind of doing the same things as bisulfite sequencing.
Yes, but you're not having to do the bisulfite treatment, which tends to be really hard. Yeah, on DNA.
And it's also worth noting that you couldn't do this with RNA though, because RNA is also modified, but you have to at least.
That's
convert it to
correct.
cDNA for PacBio, so you lose the modifications for that.
Yep, you do. It's also a higher cost than the alumina, and its competitor would be nanopore. So that would be using either a min ion or it would be using the grid ion or something like that.
Do you guys have a min ion or anything?
We have a min ion, but we do not, because you're not allowed to offer min ion as a core service. So we can play with it, but we would need the grid ion or we would need the prometh ion.
That's weird. They're weird with their
They are.
closed source stuff. Yeah.
Okay.
So
Yeah. You need to like join their cult.
you do.
I swear to God.
Okay, shall we talk about single cell? How's our time?
Oh, you're fine.
Okay, so here's my next show and tell. Yes. So most of you, most of you know that what we need to do for single cell is just get rid of noise. So the idea being that if we can look at the transcriptome of a single cell, then we're getting more information. We can identify subpopulations, okay? That if we're just doing both, they've been averaged out to some level. So these are some of the applications, are to find these rare subpopulations. To look at circulating tumor cells, for instance, to look at microenvironments, to look at stem cells. And there's 2 different ways currently that we're doing it in the lab. I sent around the cartridge for 10X, but the first one I'm going to talk about is Parse Biosciences, which is the equivalent of SplitSeq. So it's SplitSeq that's been commercialized. And they can go up to 1 million cells.
Stressing the analyst
Which
out.
would be, yes. And we are getting ready to do 1 million, just to let you know.
So awesome.
And so 1 million would be up to 96 samples. Okay. Most of the projects that we're seeing come in are doing the 100,000. Okay. And that's because price wise, the 10,000 down here, you might as well do 10X. Price wise, you're not getting much of a break. Okay. So what does SplitSeq look like? So this is what the workflow is for 100,000 cells, where you can go up to 48 samples, okay? So for one thing, sample prep wise, just to begin with, these samples are fixed. And they're fixed using Parse's fixation kit. And I think it's like $240 something maybe for 8 samples. So the beauty of that is that if you can imagine starting with 48 samples, there's no way that somebody's going to come to me with 48 samples for 10x. You just can't have them all viable. You can't have them all counted well. It's going to take you too much time to prepare to bring them to me. So for instance, if you have a time course experiment, you know, and I've seen some of those with 10x, and, you know, it's like every 24 hours, you know, can you be here available, Melanie, for this number of samples? And we'll do that. We have done that. Another way to do it is that you fix them and then you store them. And when your experiment's done and you have all your samples, then it's like, okay, now we can start the protocol. So in this case, let's just say you're bringing us 48 samples. We're into the split mode, okay? And we're going to pipette them into wells on this plate, which is supplied by Parse. And the first step is actually a reverse transcription. And within those wells, it's going to add a barcode in this reverse transcription reaction. Then we're going to take all of the cells from all of the wells, mix them all together, and replate them on the 96 well plate, and so splitting them again. And each of those wells has another barcode to add. And this addition is going to be done by ligation. So this is a ligation step. So now, theoretically, your RNA is going to have one barcode from your first number 1 step, and then it's going to have different barcode from your number 2 step. And it's going to be kind of this multi-combinatorial event, okay? Then we're going to pull them all together, split them again, and ligate on another barcode. So this is going to happen. So you've got 1, 2, 3, all the way down to 4 barcodes. So because you have these different combinations that can be, I think it can be, I'm not going to give a number. It's not good. I tried to remember that, but I'm going to get in trouble if it's a lot. Okay. So that you've added now this barcode, you go ahead and you make the library, you sequence it, and all of the RNAs from one cell should have that same sequence, okay, of barcodes. So this kind of shows you.
Wait, can I
Yeah.
ask you a quick question here about the genomics high-throughput facility specifically? Do we just need to hand you guys the fixed cells?
Yes,
You do the rest?
yes, but please
Okay.
count.
Okay.
Okay, if you count them, then that's really helpful.
No, that's excellent. No, that's good to know.
Yep. So this represents kind of what the plates look like. So for the 100,000, we're actually just using the first 4 rows of the plate, and then it's 96, 96. And in this last group, it's actually what they call a sub-library barcoding. And the nice thing about this is that we've now divided it up so that, let's just say you had one set that just didn't seem to work. You know, it hasn't, we have some level of quality control in this group. This is what the software output looks like, and it's a lot like what Cell Ranger looks like. You know, you still have your knee right here that you're looking at, sequencing saturation. It'll give you a Disney plot. And so pretty much just the same thing as what Cell Ranger will do.
And so when you're done with the barcoding, you lyse, and then you just literally, and then you put it on the aluminum.
That's correct.
Right.
We finished making the library. Yeah. And then we sequence it.
Excellent.
And it's about the same sequencing. So, okay. You know, most of these companies are going to tell you you can get by with 25,000 reads per cell. Okay. We rarely see people happy with that. If you look at the sequencing saturation curve down here, you're still a long ways from being saturated. So most of the time, sometimes people will do a more shallow run, maybe 10,000 per cell, just to get an idea of how much more sequencing do I really need. And so, but most of the time we're getting more into the 50,000 per cell range.
I have a quick question. Is the first barcode that's on there, can that tell you what sample it came from?
Yes,
So you use that one to identify the
Yes.
sample or condition?
yes, yeah.
Okay, gotcha. It's really cool.
So at scale, meaning if you're at 100,000 or 1 million, that it does, your costs are lower per cell or per nuclei. You can also do nuclei on here. And it doesn't require any specialized equipment. Okay, it's just you need somebody that's got a multi-channel pipetter to do the work. So all of these reactions are performed in 96 well plates, and the starting material is fixed, which means you can store the sample for months. The other big difference that sometimes people don't realize is that in that reverse transcription step, it's not just using oligo-DT. There's also random primers. So you're actually getting more sequence of the transcript than you are with 10X. Okay, and I'll show you on the 10X platform, okay? So this would be, and this is why they call it 3' UTR, okay? Is that we're going to have these beads down here, and this R1 is a portion of the read 1 primer binding site. Then you have this 10X barcode, which is going to be the same sequence on the entire bead. And then you have a unique molecular identifier and this oligo-DT region. Okay, and these primers are located in this bead, which is a hydrogel. So these beads and actually cells and enzyme go into that cartridge here. So we pipette them into rows. There's basically a pump that then makes an emulsion, and we take the emulsion out of the top here. So the idea is that in this emulsion, then your hydrogel beads down here dissolve. And all of these primers are released within this droplet. At the same time, your cell is lysing. So you have to have two things happening in that droplet. Okay, the hydrogel dissolves, the cell lyses, and now we've got these primers that can hybridize to the poly-A regions of your messenger RNA sequences. There's the enzyme in there, reverse transcriptase, so it's making first strand synthesis. So when people bring their single cell suspension to us, you know, if they're telling us, oh, we're going to bring it to you, you know, 7 p.m. Well, that means we're going to be there a little late, because not only do we make the emulsion, and this step is maybe 18 minutes, not very long, that's okay, but we need to get through this reverse transcription step. So we need to make that first strand, okay? And then we can store it, come back, finish library construction. So if you're looking at this, and this should look familiar to you, here's your P5 adapter, all right? Here's your read 1 primer binding site. Here's our 10X barcode and our unique molecular identifier, our poly region. Here's our insert, which is actually from read 2. And then our I7. So in this, and actually 10X now makes dual indexed adapters for us, so that we can multiplex more on a lane. And then our P7. So they're going to look at all the cells that have the 10X barcode, group those reads together, all right? And these are the RNAs, again, just looking at the 3' UTR that belong to this particular cell. We can also do ataxy. So not only can you look at gene expression, but you can look at DNA epigenetics, which parts of the DNA are actually open. And in this one, what happens is that we request that people provide us with a nuclei prep. Before it goes into this cartridge, we then use a transposase, which goes in and will only cut open areas of the chromatin. So, and it adds a sequence that's specifically for that transposase. So then we take it through the rest of the emulsion, this whole thing, and then we go ahead and we sequence it. And instead of being gene expression, we're looking at open regions of DNA in this particular protocol. Well, now we can do both within the same single cell. The difference that you need to keep in mind is that this starts with the nuclei prep. So we're losing any cytoplasmic RNA that you would have seen, you know, if we were doing whole cells. So from the,
reason why you need to start with nuclei rather than whole cells.
we can't get the transposition. Yeah.
Okay. Okay.
So
But you don't have the same problem with the reverse transcriptase getting into the nucleus if you want to also get the nuclear RNA from whole cells, correct?
so keep in mind, the cell has to lyse.
Yes.
So then the reverse transcriptase works.
Okay.
So, but keep in mind with reverse transcriptase, this is why we keep asking that the cells be washed before they're brought to us, or the nuclei be washed. If there's leftover, for instance, EDTA in there, then our reverse transcriptase is not going to work. So we need it to be super clean because we're doing some enzymatic reactions. Okay,
And, sorry.
that,
Is it, correct me if I'm wrong here, but is it true that if we're doing single nucleus, so we're losing cytoplasmic
that's correct.
RNA, but the trade-off is you can do, technically you can sequence more cells? Is that true?
Not necessarily.
Okay.
Yeah.
Okay.
Thank you. Question here, for this model, is the barcode the same, like?
Yes,
exactly the same?
yes. Yeah. Because the oligos on the surface, or it's not necessarily on the surface, the primers that are associated with the hydrogel bead, they're different primers, but they'll all have that same cell barcode. So some of the primers will be for the Nextera, which would be for the transposase portion, all right, to capture that, but they'll have that same cell barcode. Okay, and for you all that like to do analysis, you know, this is the simplified version, but basically you just take it through two different workflows, one of them for your ATAC-seq and the other one for your 3' gene expression. Okay, so these are some of the things that you think about for single cell approaches, and I'm running out of time quickly, but one of the things that I would like to show you are some of the single cell DNA that we have, okay? And we have a platform called Tapestry, and it's, again, another droplet-based technology, and that one looks kind of like this. And for this platform, you can do DNA as well as protein. And so that's why people like this. It's got 2 chambers, and that's because it makes the emulsion twice. So I'll show you that in the next slide.
And what is the protein based on? Is it?
It's a
Antibodies or?
correct. So you for phenotyping, you will get the total seq-D cocktail, and they've got a heme oncology. They have other cocktails that they can get, okay? And these cocktails have antibodies that are labeled with these oligos that are basically barcoded. So the first thing you do is you take your cell mixture, okay, and you bind it with these antibodies. And these are the different panels that they have for the DNA. This is not getting all of the DNA. It's a targeted DNA approach. So this is what the workflow looks like. We've stained the samples with antibodies. We then put them in that cartridge along with oil and a protease so that we release the DNA and the protein within the emulsion. And then we add barcoding beads in the second part of that workflow, and then we label them. So it's a 2-emulsion protocol. And what you've done now is that you've got within a single cell, okay, you have information about what proteins were associated with that single cell. And then depending on your DNA panel, you've got information about the DNA sequence of that particular gene in that panel. So some of the panels, for instance, are a myeloid panel, which is 20 genes. So we are talking about a panel. We're not talking about hundreds of genes, okay? A tumor hotspot, for instance, which is 59 genes. So some of the things that you can look at, for instance, is what happens to these, like the tumor hotspot, what happens during therapy. So we're looking at, here's initially starting at diagnosis. You would have some idea of how much of that particular mutation was present. And then during therapy, what happens to that mutation? Is that particular cell gotten rid of, eliminated, or whatever? And then potentially you could look at relapse as well. Okay, do we want to talk spatial, or do you want to just have me come back?
How about this? How about we pause for questions?
Okay,
I don't know, Sam, are there any Zoom questions?
Not currently.
Okay. Honestly, I think we might ask you that.
I'm happy to do that.
Sam, what do you think? Because we're running low on time.
Yeah, I think I would have too many questions about spatial. It would take too long.
Okay, all right. I'm happy, and I apologize. I'm just really, to be honest with you, I'm really happy to be able to explain this to
Yeah.
you guys. My dream person in the lab is somebody that can do the wet bench and the bioinformatics. I just feel like it really adds to the understanding of what it is that you're doing. And so to be able to come in and to talk to you about it, I really appreciate the opportunity.
Melanie, I can tell you, this is probably one of the most important talks I've ever had.
Well, thank you.
I really wish I'd seen this talk like 2 years ago or 3 years ago when I started. I have such a better understanding of what's going on now. This is truly amazing. Thank you.
Thank you for inviting me.
Do we have other questions, guys?
Okay, let's find a time,
Yeah.
okay, that's convenient, and I'll come back with spatial.
Probably have to be in the fall.
I'm fine with that. There'll be all kinds of changes by the
Yeah,
fall.
sure.
Excellent.
It's just constantly ongoing,
Yeah.
especially in the spatial area.
I know they keep making the spots smaller, so.
They are, yeah. It's the Wild West out there in spatial, so.
Great.
Okay,
All right. Well, thank you, Melanie, for.
thank you all. Thank you very much.
We hope to see you again soon. Yeah, that was amazing. I mean, I really wish I'd given that talk 3 years ago.